Perfect. So I'll start with a brief introduction to why we need to do these things, and then start with talking about normalization, a bit about gene set selection, and removal of confounding factors. So we talked a lot about it during the QC lab lecture and so on, that we have different sources of variation in our data. We have biological variation, like different cell types, different cell states, cell cycle, cell size, sex, age. I mean, you can think of a lot of different things that can be biological variation in a data set. And then I like to distinguish technical variation. That's more due to the quality of the cell, the different library prep, batch effects between the different samples. And ideally, we want to remove all of these other sources of variation when we want to identify cell types. We want the cell type signal to be as strong as possible and not these other things. Of course, if we're looking at and studying cell cycle specifically, we might not want to remove that one and so on. So it depends on the question you're after to really think about what's the main variation in the data I am interested in and want to really enrich for. And this is not an easy task, I must say, because we can tweak our data quite a lot to try to remove all other sources of variation, but there's always a risk of also removing the interesting variation that you want to keep. And UMis solves some of the problems with technical variation, but not all of them. So this is just a table from another paper where we already talked about this, that we have a lot of cell-specific effects and a lot of gene-specific effects that will vary between genes and samples. And it's pretty much just gene length and amplification bias that UMis will take care of. All of these other issues still remain. And we can pretty much say that we have three types of normalizations, the three main types of normalizations, either just normalizing by total counts, or also include gene length normalization, and that mainly, of course, applies to full length methods. If we have these tag-based methods, gene length doesn't matter. And dropout rate normalizations, where we try to estimate dropout rate per cell and normalize by that instead. And I think pretty much everyone here has worked with bulk RNA-seq and know that there's a lot of different ways of doing normalization there, either just counts per million or normalizing by gene length in RPKM or FPKM or TPM. So I'm not going to go into detail with these ones. There's several additional ways of doing it where you try to improve the idea of like what group of genes you use for normalization, where the extreme outliers and so on are not driving your normalization as much. And some of these actually work fairly well for single cell RNA-seq. Especially VOOM has been shown in some, Limma-VOOM has been shown in some of these evaluation papers that it works quite well for single cell RNA-seq data. But then there's a long list of papers published with different methods to normalize single cell RNA-seq data. And I won't go through all of them. I will talk about a few of them. And then it's up to you to judge what makes sense. It's also important to think about that we pretty much always work in log transformed space when we work with single cell RNA-seq data. It's also the same for bulk data. With bulk data, it approaches a normal distribution when you do log transformation, while some people claim it's a zero inflated negative binomial, it should say. I wrote this too fast. And so the non-log transform here is just one of the cells from from this COVID data set, where you can see the extreme differences between like the highest expressed to the next highest expressed. There's just a few genes here and there. Compared to if you log transform it, you get a more even distribution of the data. And in practice, I mean, this is a very simple operation. You divide it by the sequencing depth per cell. You multiply it. Normally, I think the scale factor is 10,000 in most of these packages, since that's about the sequencing depth you will have in most 10x experiments. But it's also good to think about, sorry, that if you have, for instance, SmartSeq data or very deeply sequenced data and so on, you might need to adjust your scale factor for that data set. And in Skater, the function normalize, you can either just use total counts, or you can provide individual size factors per cell. And it log transforms in the same step. The same for Seurat normalized data. It log transforms and normalizes in one step. In ScanPy, it's two separate functions for normalization and log transformation. So if we do depth normalization, we're kind of assuming that we have about the same RNA content in all cells. And that works well in homogeneous cell populations. But in most cases, it differs quite a lot between cells. And also something to think about is these outlier genes that takes up a large proportion of the reads. They will have a big impact if you have one gene taking up 50% of all your reads, and then you just simply divide by total counts, that all the other genes will have a very low expression. So one strategy is this deconvolution method that's implemented in the ScRAM package. It's basically just, they call it pooling across cells. So they create these random sets of cells and compare the sum of counts for this set versus the sum of counts of all cells. And they iterate over this multiple times to calculate the size factor for each cell. And actually, here is just an example, especially for small cells. Here is, I think it is, this is the true median, and this is the shifted median that they estimate from this deconvolution. And this can be run in ScRAM, either just with random sets of cells within your whole data set, or you run clustering first, and then you do this where you do these pooling just within each cluster separately. And Another way of doing normalization is to group the genes, because global scale factors.
I'm sorry,
yeah.
can I ask
yeah.
a question? So
Yeah,
if, for instance, if you do an overexpression experiment, it's going to be like crazy highly expressed, right? Wouldn't it be better to just remove this gene then from the data set?
That depends on the gene, of course. Most overexpression experiments will maybe be like, if you take a transcription factor that regulates something and you overexpress it, it goes from low to medium expression, perhaps. So it clearly depends on the gene in question.
But what is the threshold where you have to take into account when you do normalization then?
That's hard to say. I'm always worried and try to figure out if it's biological or not, if I have genes that are more than like 20% or something, I would say. 10 to 20%. If it's over 50 for sure, that will skew normalization quite a lot.
Yes, unless you're doing overexpression of ribosomal proteins or mitochondrial or something, then that will be quite a problem. But for other genes, I don't think they would contribute that much to the overall pool of genes, especially because most of the genes are very lowly expressed compared to the bulk of, let's say, ribosomal genes.
Yeah, I mean, it clearly depends on the cell type also. In some cell types, you might have actin extremely highly expressed, taking up. And if you would overexpress that, you would pretty much only have actin in the cells. I don't know if they would, how that would work. So it's really thinking about the system you have. It's also a case where even though it's a gene you're interested in, you can remove it from the normalization for doing clustering and everything. And then you can bring it back again and do differential expression with it and without it and compare the results and see. So it clearly depends on what your question is and what those genes are. So there are some methods where they try to instead bin the gene by expression level. And one of this, the first one that came out is SCNorm. So basically what it does is to bin genes by gene expression. So you take the lowly expressed genes and you take the highly expressed genes and the intermediate in different bins instead. Because they will have very different distributions if you take for these different levels. I can show it maybe better here. So they basically do a quantile regression for each of these levels. And what they show with just doing normal types of normalizations, like I'm not even sure what MR is right now, but the lowly expressed genes will tend to have negative fold changes in this comparative simulation, even though it's supposed to be a no change case, mainly because these ones are driving but it's also the biggest group of genes. So I don't know how they bin it exactly. So they show that you can use this to get decent level at all expression levels instead. And similarly, with a very similar idea, Seurat uses SCTransform. It's developed by the same lab as the Seurat package. It's more or less the same. They bin the genes into different groups. And then basically, instead of negative binomial regression, they do, it's just a different regression method. I think I've done some comparison of the results, and it looks fairly similar if you do the SCNorm or if you do the SCTransform. The whole idea is very similar. What is also really clear when you do this is that at the same time as you do this normalization of the genes, you remove the effect of like number of counts and number of detected genes per cell, because then the scaling of the genes will not be as affected by how many detected genes you have per cell. And if you run it in Seurat, it actually does variable gene selection in the same step. And you can also add regression factors in the same step. So if you want to regress out certain factors. Then there's a bunch of different methods. You can use SimbaWave. It's both gene level and sample level things. So you can add in different like number of mitochondrial. You can add in batch sample batch effects and all this stuff at the same time as you do this normalization of the genes. I have never tried this method. I don't know if you have, Paulo.
Yes, but it's very, very slow. So it's really, it's a quite good model if you have small sample size, like for example, for this tutorial, like 10,000 cells, for example. But if you have too many cells, since it's tried to fit a regression model and matrix factorization, it becomes just too slow for bigger data sets. And I'll also mention that in the data integration, because that's also a way of doing data integration. And it also reduces the space to PCA in the end. That I'll also mention later.
Yes, exactly. It works on the PCA instead of on the gene space. And here's just a comparison. So here is just a summary from one of these comparison papers where the size factors end up being very different depending on what method you use. But what's more important is that it has actually, and also in this paper, in my experience, what normalization you do doesn't have a big impact on clustering, dimensionality reduction, and so on. But it has a big impact on differential expression, or big impact. So basically here they've done simulated data where they either have sort of a symmetric case where you have equal number of up and down regulated genes in both directions between two clusters, or if it starts being asymmetric or completely asymmetric, where you only have upregulated genes in one cluster, and then the proportion of differential expressed genes. And as you can see, if you only have upregulated genes in one cluster, and most of the genes are upregulated, pretty much all of these methods fail, regardless of how you normalize it. So in summary, normalization doesn't matter that much for clustering, but has a big impact on differential expression. So especially when you're trying to compare very similar cell types, where it's hard, where you don't get that many differential expressed genes, or you get weird differential expressed genes, you might need to go back and think, should I do some size factor normalization? Should I do something different when I normalize my data? So as I said, normalization has a big impact. on differential gene expression and not as much on clustering. In most cases, it's quite fine to just do the sequence depth normalization. I think it's mainly when you have very similar subtypes or cell types with extremely different cell sizes. It might be good to have these individual size factors. And doing these kind of SCTransform normalizations also helps to remove the gene detection effect.